Cek.
Agak delay sih.
Cek, cek.
Emang delay kalau YouTube. Ya kalau YouTube mah delay. Iya.
berlokasi di Tes. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. The Honorable Head of Department Cardiology and Vascular Medicine, FMUI, Dr. Bambang Widiantoro, SPJP, PhD. The Honorable Head or Representative of Cluster Metabolic Disorders, Cardiovascular and Aging, IMERI, FMUI. The Excellency, our speaker from Kobe University, Professor Noriaki Emoto, MD, PhD. And all of distinguished participants who come to IMERI today, and also participants who join via YouTube. On behalf of the Head of Magister Program in Biomedical Sciences FMUI, Professor Wawai Muli Arozal, who was unable to attend this event today, I am Rani Wardani Hakim, would like to welcome all of you to our guest lecture session with the title Bringing Endothelin from Peptide to Treatment, a Successful Story of Translational Research. This event is collaboration program between Magister Program in Biomedical Sciences FMUI, Department of Cardiology and Vascular Medicine FMUI, and also Cluster of Metabolic Disorders, Cardiovascular and Aging, IMERI, FMUI. Today we are excited to have Professor Noriaki Emoto with us, who is an expert in cardiology and vascular biology, and also the adjunct professor of Universitas Indonesia since 2017. Good afternoon, Prof. Thank you for coming to us. Professor Emoto will share to us the journey of endothelin research from a simple peptide to a groundbreaking treatment. We will hear from Prof. Emoto about the challenges, the breakthrough, and the impact of the endothelin research on the medical field. This lecture will lead by our distinguished moderator, Dr. Hari Sakti Mulyawan, PhD, SPJP. Good afternoon, Doc. Thank you for coming. Before we start, I would like to read the CV of Dr. Hari as follows. Dr. Hari Mulyawan, MD, PhD, born in Jakarta, 1st October 1984. He finished doctor program in Cardiovascular Science, Kobe University Graduate School of Medicine, Japan, from 2011 until 2015. He is a cardiologist in Department of Cardiology and Vascular Medicine, Faculty of Medicine, Universitas Indonesia. He did his postdoctoral research at Kobe Pharmaceutical University, Japan in 2015. He is now head of scientific program on Indonesia Pulmonary Hypertension Working Group. He has received Young Investigator Award about Endothelin 1 in 14th meeting Savannah in Georgia 2015. He is now a research coordinator of Department of Cardiology and Vascular Medicine, Faculty of Medicine, Universitas Indonesia. Without waiting any longer, we will please Dr. Hari and Prof. Emoto to come to the stage and to start the lecture. The stage is yours. Please come, Prof. and Doctor.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. It will my honor to introduce my mentor, my supervisor, Professor Noriaki Emoto. His birthplace is in Osaka, August 31, in 1961, and he is a postdoctoral fellowship under supervision Dr. Masashi Nagisawa, one of the founder of the Endothelin, right, in Dallas, Texas, and Professor Noriaki Emoto won a lot of prize, won the Louis Katz Basic Science Research Prize of the American Heart Association in 1994, and he is also for the founder of molecular identification and characterization of endothelin converting enzyme. So follow from the Yanagisawa Sensei as the founder of endothelin 1, and then Professor Noriaki Emoto found the endothelin converting enzyme 1 and 2, and he is an advisory board member of Endothelin International, director board member of Japanese Pulmonary Circulation and Pulmonary Hypertension Society, and he also as the chairman twice in Endothelin 13 held in Tokyo, and also the Endothelin 16 held in Kobe in 2019. And since 2017, he already become adjunct professor in University of Indonesia, and but since COVID, yeah, he cannot come to Indonesia. So it is our great honor to have you again in here to give lecture regarding basic translational, regarding this journey from endothelin 1 into groundbreaking therapy. Professor Noriaki Emoto, the time is yours, please.
Thank you very much, Dr. Hari, for your kind introduction. And good afternoon, ladies and gentlemen. My name is Emoto, and as Dr. Hari introduced me, the last time I came here was 5 years ago, 2019, November, here. And after that, because of the COVID-19 pandemic, I cannot come to this country. So I'm very happy to return here to the beautiful country, and I have a chance to see many of my friends, teachers, and colleagues. So today, I would like to talk about the endothelin system. And okay, so before talking about the endothelin system, I would like to explain about drug development. So it's, yeah, because I initially graduated from the medical school. I completed the residency as a cardiologist. And at the time, I was wondering what I should do, work as a physician, and of course, this is the one choice, and another choice was to do some research so that I can contribute more medicine as compared with I work as a physician. So I chose to be a researcher or scientist. So I joined Professor Masashi Yanagisawa and pursue that kind of drug development. At the time, I dreamed that I can contribute to the drug development. And it turns out not that easy. So I would like to talk about that. So as you can see here, drug development takes a long journey, starting from the isolation of the molecule. And we have to characterize a lot if what kind of disease would be a target. And once we found the target, we have to do preclinical study using mice or rat or monkey or pig. And if it's okay, then we're going to apply to the human. So, yes, please. Could you please? Yes. So, usually it takes 9 to 17 years that since one molecule was identified and to be applied to the drug. Nowadays, availability of AI or some sophisticated method, this term would be shortened. But previously, it took more than 10 years. And please. Yes. So the probability to be success is very small. Yes, please. Yes. It's about 1 in 25,000 chance. So it's like you can get lottery. And yes, please. So in the case of endothelin, please, yes. And here, yes. So this is the very, very successful. And finally, it turns out to be a very effective drug for pulmonary arterial hypertension. Okay, so then I would like to talk about a little bit history. So please just relax and listen. I would not give you an exam, so just listen. Okay, so endothelial cell, vascular endothelial cells, is one layer of the inner side of the best cell, and which was thought that just to one structure of the best cell. However, these two preparations, by using these two preparations, one is intact endothelial cells and another one is disturbed endothelial cells. Please. The behavior of the best cells were different. So this is found by Dr. Forchigot. So if endothelial cells are intact, then if you add acetylcholine, then best cell will dilate it. But if the endothelial cell disturbed, then those acetylcholine-induced dilatation would disappear. And next, please. And next, please. So because of those, those factor was turned out to be nitric oxide, NO, and which, so those three doctors were awarded as Nobel Prize, Physiology or Medicine in 1998. So this is 1980th that they are actively working. So many people were trying to find what's this mysterious factor which causes the vasodilatation secreted from endothelial cells. And please. So around that time, this is my mentor, Dr. Masashi Yanagisawa, when he was a third-year graduate student. So he found that some factor are secreted from cultured endothelial cells, and this, when he put this supernatant, then best cell constrict, so not dilated, constrict very strongly. And he finally purified this enzyme, purified this peptide, and turned out to be 21 amino acid peptide, which is later named as endothelin 1. And next, please. And published in Nature, a Nature article, not letter. So it's kind of astonishing manuscript in 1988. That was the time that he has not graduated from graduate school as a student. And I can tell you how astonishing it is. So they sent this paper to the Nature, and at the time, chief wing editor is Sir John Bain, who is also a Nobel laureate, and he was a chief wing editor of Nature. And the staff put this manuscript on his desk. And when he saw this one, he was so astonished, but he realized, oh, today is April 1st. So this is a very good joke of April 1st. So he believed that this is joke, but actually this is not joke, the real manuscript. Okay, next please. Okay, next please, and next please. So endothelin 1, 21 amino acid peptides, shows the potent vasoconstrictive activity, and also when they inject into the rat, then this rat shows very high blood pressure and very long sustained high blood pressure increase. So this potent blood pressure elevation effect is much more than catecholamine. Okay, and next please. One more, yes. So many people were thought that, oh, this would be related to some cardiovascular disease, and also this could be a target of a drug. So next, please. And later on, based on the structure of this ET1, isoform of ET1 ET2 and ET3 were identified. So this is called the endothelin families. And next, please. And next, please. So this endothelin acts on two isoforms of G protein coupled receptor called ETA receptor, and ETB receptor shows a variety of biological actions, including vasoconstriction or vasodilatation. And next, please. And this endothelin 1 is produced from precursor, and this precursor have no activity called big ET1 by endothelin converting enzyme. This is analogous as angiotensin system. So angiotensin 1 is cleaved and produced angiotensin 2 by the cleavage of angiotensin converting enzyme. And next, please. And next, please. And next, please. One more, please. And one more. And one more. Yes. So as a graduate student, I have been working to isolate this enzyme, and finally, I succeed to purify the enzyme from bovine adrenal glands. Why bovine? Because we were at the time in Texas, Dallas. And you know, Texas, there are so many cows or bovines, so that we can easily get adrenal gland from bovine. And next, please. So, the next what I did in our laboratory, we working to find the pathophysiological or physiological role of endothelin system. And one of the methods is to create or generate gene-modified mice. And in this case, these mice lack ECE1 gene or ECE1 gene deleted mice. And this one is ETA receptor deleted mice. So these two genetically modified mouse shows a very similar phenotype. And for example, as you can see, the right side and as compared with left side is a normal, the glutamate. But as compared with this normal, this knockout mouse, the color of knockout mouse is rather white, which means this is cyanotic. So this mice cannot breathe very well because they have a retarded jaw, the abnormality of the jaw. And next, please. In addition, these mice shows the congestive heart failure as before birth. And this congestive heart failure is caused by the structural abnormality of the heart and big vessels like pulmonary artery or aorta. And next, please. So these mice shows abnormality of jaw structure and the cardiac and the big vessel structure. That means those parts are developed from first pharyngeal arch, which is derived from neural crest. So this phenotype suggests that endothelin system is important for the development of neural crest-derived cells. And next, please. Then, what's the other isoforms? For example, this is ETB receptor knockout mice or ET3 receptor knockout mice. Both shows the same phenotype. One feature is white spotting. So these mice supposed to be brown, but these mice shows the white coat color. And these mice would die within 3 weeks because of the very dilated colon, which called toxic megacolon. Those phenotype was really identical a human disease called Hirschsprung disease. And next, please. Hirschsprung disease is caused by the lack of enteric neuron, and enteric neuron is derived from neural crest. And skin color also, the melanocyte, the number of melanocyte does matter, but melanocyte is also derived from neural crest cell. So from this, ET3, ETB are also important for the neural crest cell derived development. And next, please. So these ET1, ETA, or ET3, ETB axis turned out to be important for the development. And next, please. Yes, and the next, please. So how about human? And actually, there is a disease called Shah-Wallenberg syndrome. So this syndrome present Hirschsprung disease and skin color abnormality. And please, yes, so this girl has that disease. It's difficult to see, but this eye color is different, brown and blue. And this is white spotting the skin. And she has ETB receptor mutation. And there is family, which suffered from same abnormality. So also in human, ET3 or ETB are important for neural crest derived cell development. And next, please. So that was a big finding or discovery, but my initial intention to start research is to find something which is contribute to the medicine, especially cardiovascular disease. But this is nothing to do with cardiovascular disease. So yeah, I was so disappointed, and I was wondering what I should do. So at the time, pharmaceutical company tried to develop the antagonists for these receptors, and the first successful antagonist is, could you back? Yes. The most successful antagonist is called later bosentan, or brand name is Traclia. So this is the first publication about that drug. This is actually compound at the time. And as you can see, this compound was published in Nature. So that means there is a huge impact that could be a drug for cardiovascular diseases. And next, please. So after very successful data for preclinical disease model, this drug and many endothelin receptor antagonists were used for clinical trial, including hypertension, heart failure, and cancer or autoimmune disease. But unfortunately, hypertension or heart failure, although the preclinical data was very promising, the human trial failed, so that they are not approved as a drug. Next, please. So for example, this is very strong vasoconstrictor. Why antagonist does not work? And next, please. And next, please. So we try to figure out why. And this is the data from Professor Kurihara from Tokyo University, and they used endothelin heterozygous mice. These mice have half amount of ET1 gene. So when they check the amount of endothelin 1 in serum or lung, those heterozygous mice shows about half. And next, please. And they measure the blood pressure. So the amount of endothelin 1 is half. Then they or we expect the decrease of blood pressure. And next, please. And next, please. But unexpectedly, blood pressure was increased. So that was, again, caused a big debate. Many people sent a letter to Nature that that could not happen. And next, please. So we tried to figure out the reason. And at this time, we can generate the cell-specific gene deficient mice. In this case, we developed or generated vascular endothelial cell-specific ET1 knockout mice. And next, please. So this mice shows almost no ET1 in the serum. And next, please. But they can survive. And at a glance, it's normal and are normally developed. So we use these mice for studying about the physiological role of endothelial cell-derived ET1. And when we measure the blood pressure of these mice as compared with the normal retamate, blood pressure decreased. So from this data, we concluded that ET1 is important to maintain the blood pressure. Then why half amount of ET1 does not decrease the blood pressure? And next, please. And next, please. And one more, please. Yes. So from our data, endothelial cell-derived ET1 acts on ETA receptor on vascular smooth muscle cells, cause vasoconstriction or vascular remodeling. That is for sure. But the story is not that simple. And next, please. Yes. And next, please. So ET1 is ubiquitously expressed. This is the old data, so-called northern blot, to see the mRNA amount of the each gene. And as you can see, ET1 is highly expressed in the kidney. And next, please. And this is another study done by Cheryl Garpy. She checked the ETB deficient rats. And this rat shows the, usually without any intervention, the normal blood pressure. But when these mice subject to high salt diet, then blood pressure increased. And next, please. And when she used, for example, calcium antagonist or ACE inhibitor, it does not work. And please, next, please. Only amyloid can decrease, next, please, the blood pressure. This amyloid is a drug for a disease called Riddle syndrome, which is very rare hypertension causative gene. And next, please. So ETB deficiency appears to be acts on the sodium handling. And next, please. And next, please. And one more, please. And one more, please. So I will skip these slides to make long story short. We made many kinds of knockout mice. And next, please. Next, please. And next, please. And next, please. Yes. So in the kidney, ET1 produced from collecting ducts, cell of collecting ducts, acts on ETB receptor to suppress the salt and water retention. And next, please. So the blood pressure regulation is not only the vasoconstriction, but also the water retention or salt retention. So endothelin work, in a sense, blood pressure elevation, but in another sense, blood pressure lowering effect. So the balance of those may regulate the blood pressure. So anyway, endothelin receptor antagonist does not applied or approved for the blood pressure lowering drug. Then what's next? And this is another disease called pulmonary arterial hypertension. So could you back? Yes. So this is the very typical histological structure of pulmonary arterial hypertension, which is called plexiform region. And this is the very difficult disease. The mortality rate is very high, especially the young female suffered from this disease. For my case, my patient is like 17 years old or 18 years old or 22 years old. So those girls, once suffered from this disease, would die usually within half a year. Very difficult disease. And this histological data shows that this typical region, endothelin and endothelin receptors are highly expressed. And next, please. And as I told you, this survival rate is like 5 years survival rate is almost 30%. So this is much worse as compared with, for example, stage 4 malignant cancer. And next, please. But this endothelin receptor antagonist was very effective for preclinical disease model. Then the clinical trial started, and as you can see here, the exercise capacity measured by 6 minutes walk distance improved dramatically in dose-dependent manner. And next, please. And actually, the survival rate of those patients are improved by endothelin receptor antagonist. And next, please. And next, please. And next, please. So currently, we have 3 kinds of endothelin receptor antagonists, ETA selective antagonist or both ETA-B dual antagonists. And next, please. And by using those, and not only endothelin antagonist, but also NO, the PDE5 inhibitors or prostacyclin analogs, those 3 drugs are very effective for the pulmonary arterial hypertension. And next, please. So now this is the current data in Japan. So 3 years survival rate is 95.7% because of this drug. Okay, so when we referred the pulmonary arterial hypertension patients, like the age of 20th, and we used to explain the family or the patient that there is no way to treat this disease. So I'm sorry, but your life expectancy is like 3 months or 6 months. But nowadays, we can say that, okay, if you use drug, then more than 90% you can survive for 3 years. Of course, not cure, but at least they would not die. Okay, and next, please. So then why endothelin receptor antagonist is effective? Not effective for systemic hypertension, but effective for PAH. And next, please. So again, coming back, this mice, we use this mice for several disease model to generate disease model and try to identify the role. And next, please. And you may see this guy. This is Dr. Bambam Widiantoro when he was a graduate student at Kobe University. And do you know what's he doing here? He's taking echo echocardiography of mice, yes. He took echo of mice, and of course, many things he did. And finally, next please, he found that the endothelin is related to the cardiac fibrosis, and which was published in Circulation. And next, please. And yeah, I do not have enough time to introduce all the data, but I just summarize here that endothelin one is important for vasoconstriction or spasm, but please, next, please. Rather important fibrosis or inflammation or anti-apoptosis. And those are really related to the vascular remodeling observed in pulmonary arterial hypertension. And next, please. So probably, this is our hypothesis that vasodilation of pulmonary artery by endothelin receptor antagonist is not contribute to the improvement of the disease, rather suppression of the inflammation or fibrosis or vascular remodeling is important. So next target, so pulmonary arterial hypertension is okay. The next target is the kidney. And again, you may find this guy. This guy is also Dr. Bambam Widyantoro. And this guy, this tall guy, is Nicolas, who is from France. And they are working with the diabetic nephropathy model. And again, could you back? I would not go into the detail, but anyway, when ET1 knocked out, deleted them, the proteinuria or albuminuria caused by diabetic model are suppressed. So ET1, ETA appears to be important for proteinuria or albuminuria in diabetes. And next, please. So there are several clinical studies, and finally, this study, atrasentan, this is ETA selective antagonist, was used for diabetic model. And these are primary endpoint. And next, please. And next, please. So this study shows the ETA selective antagonist appears to be effective for diabetic nephropathy. So it's still ongoing study, and we expect that that would be clinically applied within 2 or 3 years. And this is another compound, which is one compound shows ETA receptor antagonist as well as AT1, angiotensin 1 receptor antagonist. And next, please. And the clinical trial using this compound shows the significant efficacy for kidney disease called FSGS, which is a very difficult disease for the kidney. So although it's not yet approved, there is a possibility that sparsentan, ETA selective antagonist, could be approved for this disease. And next, please. And this is another story. This is the after subarachnoidal hemorrhage, the vasoconstriction of the cerebral artery determine the prognosis of those patients. And endothelin receptor antagonists are very effective to protect vasoconstriction and contribute to the final outcome. So it's now approved in Japan. So many of the brain surgeons use endothelin receptor antagonist, in this case clazosentan, after the operation of subarachnoidal hemorrhage. And next, please. Okay, so now endothelin receptor antagonist is used for pulmonary arterial hypertension, and I didn't explain, but another approved disease is resistant hypertension, and digital ulcer, which is caused by the systemic sclerosis patients, and this subarachnoidal hemorrhage. What's next? And please, and please. And please. Okay, one more please. So, as I explained you, that ET1, ETA axis is important for the structure of jaw and vessel and heart. And ET3, ETB axis appears to be important for the pigmentation or the melanocyte development and enteric neuron development. So what's ET2 doing? So this has been a long mystery. So actually, we had ET2 knockout mice back in 1994, but still, we don't know what happened. And next, please. And next, please. And next, please. Okay, so when ET2 knocked out, then mouse size becomes small. And those mice die at longest 3 weeks. And this shows the low body temperature. And they shows hypoglycemia and high ketone. So we don't know why this happened. And next, please. So now in our laboratory, 4 Indonesian researchers are tackling this question. And this is Ahmad Mustafa, and this is Mega. Both are from Gajah Mada University, from Jogja. And they found that those mice shows the abnormality of the lung structure. And as compared with normal ET2 knockout mice or ET2 inducible knockout mice. So when in the adult, they delay to ET2 knockout mouse. This shows the disruption of the structure of the lung, which it look like the emphysema or so in the case of neonates, it's called BPD, bronchopulmonary dysplasia. And now they use a sophisticated method, including single-cell RNA sequencing, try to figure out what's the reason to show this phenotype. And next, please. And this is Indy, again from Jogja. She is working about the adipose tissue. So when ET2 knocked out in the adult food, then those mice get skinny. Lose white adipose tissue and almost gone. So when you knock out ET2, then that would be very good to lose your weight. So yeah, Bamba-san, you may need, and Hari-san also. But yeah, of course, this is pathologic feature. And this is Satorio from, again, the Jogja. And he is working about skin. And this ET2, when ET2 knocked out, then after shaving, the regrow of the skin are disturbed. So ET2 may appear for the hair development also. If we identify the reason, then we may get fortune, Yeah, and next, please. So in our laboratory, currently 8 Indonesian researchers are working to figure out not only ET2, but also several projects are very hard for this. And next, please. And yeah, I'd like to acknowledge that, yeah, Dr. Bamba Widiantoro, Dr. Skoadiaut, and Dr. Sunu were working very hard, and they got the award from Japanese Circulation Society in the past. Okay, and next, please. So these are the members from our laboratory, all Indonesian students, and who complete PhD study and got PhD from Kobe University. And yeah, next please, and one more please. So this time, I'm very happy to return here that many of alumni gather, and we had a very good time to talk about the past. Okay. Okay, thank you very much for your attention.
Yeah, thank you very much, Professor Noriaki Emoto. Such inspiring lecture regarding when you first found the molecule, and actually so many laborious procedure and laborious to get when you find only the peptide, until then you get the disease, the clinical disease, and you found and then you start to found the indication what kind of use of this endothelin. So I'm sure the audience have a lot of questions, or they're still confused. But it's okay. I think this inspiring story, I think, can inspire you to know what is the procedure for, what's the process from founding a new peptide until become potential therapy and potential disease modified agents. So, is there any questions from the audience? Any comments, questions? Yes, please. Please state your name.
Thank you, Prof, for the lecture. My name is Ray. It was very interesting. I actually had an experience as well in the translational research. I experimented with zebrafish with gene knockout as well. Perhaps I want to ask a bit about the endothelin subtypes. You mentioned there are 3 endothelin subtypes in the mice. Compared to the human, is there similar subtypes of endothelin in the humans? And then when the mice were knocked out, you mentioned there are several neurocrest-derived malproliferation. When given as a drug for adult phase, would that still give rise to some trouble in the adult stages? I think that would be my question for now. Thank you very much.
Okay, thank you very much for your question. And that is very, very important question. About the first question, the isoform of ET1 to 3, those are really preserved from fish to the human. So human has ET1, ET2, and ET3. And so this would be a very important molecule, so that across the species are preserved, conserved. And one interesting thing is some special snake in Israel, they have ET1, ET2, ET3, and another endothelin, which is called sulfotoxin. This is special toxin that snake has. So when that snake bite, I don't know, mice or something, then those peptides goes to that victim, and they died because of the high blood pressure. So some specific species may develop endothelin system for their kind of the toxin. But otherwise, ET1, 2, 3 are all preserved. And the second question, so when endothelin system are knocked out, they have the abnormality. And when, so naturally thinking that if you use endothelin receptor antagonist for pregnant mother, then that would be a big problem. And actually, it's already proven that the fetus or baby shows such kind of abnormality. But in the case of adult, it's already completed. I mean, the development has been completed. So far, there is no report about such kind of abnormality. But again, I mentioned your question is very important because so far, ET1, I mean, the adverse effect by ET1 knockout or adverse effect of ET3 suppression has not been reported. But since we do not know the ET2 function, and as I show you, when ET2 are blocked, deleted in the adult food, they shows the structural abnormality of the lung. And there is a possibility that endothelin receptor antagonist may cause some lung disturbance. And actually, pulmonary arterial hypertension are caused by several reasons. One of the reasons is lung disease, like emphysema or chronic obstructive lung disease. And endothelin receptor antagonists are not approved for pulmonary hypertension associated with lung disease. So far, no one knows. There is no evidence or no one knows the reason why it would not work. But I suspect that when endothelin receptor antagonists block ET1, but at the same time ET2, so those might be due to the consequence of ET2 blockade. If this hypothesis is true, then if we can produce or compound which specifically block ET1, but not ET2, then we may use this drug for the pulmonary hypertension caused by lung disease. So that's what we are now working. Yeah, thank you.
Thank you very much for the questions. Is there any more questions or comments? So perhaps if not, I also want to ask some questions. It's very interesting about the ET1 contribution in the lung disease and can cause by deleting or altering the ET2 can induce emphysematous. One of the common cause for emphysematous is smoking, let's say. So have you done any other experiment actually seek the role of smoking with the alteration in endothelin too?
Yes, again, this is very important question. So the expression, so long time people ignore the presence of ET2 because when you check the expression of ET2, it's very, very low. ET1 is highly expressed, but ET2, almost no expression. But we happen to find that the hypoxia and return to normoxia, suddenly ET2 expression is more than 100 times. So just hypoxia would not cause hypoxia and normoxia. So if you smoke, obviously those the vesicle should be hypoxic. And after stop the smoking, then normal oxygen comes. So I think that ET2 do something for preventing or protesting against smoking. So, we tried to figure out the effect of smoking about ET2 function, but when we use mice, smoking model is very difficult. Yeah. There is some apparatus that puts tobacco and put mice in the smoke. But it's very expensive, and we have to care about smoking, so we did not do that. But I think obviously the smoking and ET2 expression may have something. Yeah.
Yeah, interesting. So yeah, if there anybody wants to try and to seek the expression of ET1, I think you can detect the ET2 from serums, is it? From the serum, or we have to take, this is a tissue expression.
Yes, again, the difficult part is endothelin consists of 21 amino acids. And ET1 and ET2, the difference are only 2 amino acids. So currently available antibody, we cannot distinguish ET1 or ET2. But people, present that serum concentration of ET1, but strictly speaking, that's not true. Serum concentration of ET1 or ET2. This is the right expression. But as I told you, in the serum, people believe that most of the peptide is ET1 because ET2 expression is very low. But we don't know. Of course, you can distinguish by mRNA expression. mRNA, we can detect 2 amino acid differences or 6 nucleotide differences very easily. But the question is, the actual peptide concentration, nobody knows.
Okay, any more comment or suggestion maybe? Maybe from Dr. Rani or Dr. Bambang or Dr. Rani? Do you have any more other comments?
Thank you. Sensei, it's a very interesting journey of your research, and I think it's very huge impact to our medicinal field. And I think our question is represent our student here. There are many student, magister and also specialists. And then maybe in the heart they said that, wow, it's a cool research. But I think they want to, if they want to join with your team to the research in Kobe University, how to prepare for it or how to contact you. I saw from your PPT that there are many UGM students come from there, and then I think our student is too shy to ask that they are interested to join the research with you, or just maybe starting collaboration here and then assist by you there, or I hope that they can join with your team in the future. How can they do that?
Okay, thank you. Most of the Indonesian researchers are graduate students supported by Japanese government, Monbu Kagakusho or JSPS. So if you can pass the competition to get those grants, then that's the best way, because Japanese government will provide the airfare and every life expense and tuition fee. And of course, Dr. Bambang or Dr. Hari knows very well. And unfortunately, I'm too old. So since I will retire 2.5 years later, so I cannot accept new students. So this year, last October, would be the Dr. Saiful Hidayat from Gajah Mada University would be my last graduate students. Yes, but although I retired from Kobe University, I still work for Kobe Pharmaceutical University as a researcher. And although I cannot accept graduate students, but I'm happy to work together. So if we have a good collaboration, or just visit our facilities. Currently, 8 Indonesians are there, so they could help in many ways. I mean, not only research or experiments, but also life in Japan.
Okay, thank you very much, Sensei. Is there another question? Come on, students. Cukup. Yeah.
Yes, please. This is a rare experience, I think. Yeah.
Good afternoon, dear professors. Thank you for your lecture. That was a very interesting and very comprehensive research from you. I want to ask you if there is any side effects that will come with using bosentan, and whether there is any long-term side effects from that. Thank you.
Thank you very much. In the case of bosentan, the most the frequent adverse effect is lung dysfunction. So maybe 10% of the patient shows the elevation of AST and ALT. And if it's more than 100, then we have to stop the use of bosentan. And for example, the ambrisentan, another endothelin receptor antagonist, this is ETA selective antagonist, have almost no lung dysfunction. So if we use bosentan and find the lung abnormality of lung function, abnormality of lung functional test, then we switch from bosentan to ambrisentan. But currently, macitentan, this is the most recent one, and this one, almost no liver dysfunction. So in terms of liver dysfunction, if we use macitentan or ambrisentan, that's okay. And other side effect is the anemia. So the reason was not clear, but some patients, a very small portion of the patient shows the anemia. In this case, especially if the patient suffered from pulmonary arterial hypertension, anemia could cause could cause a worsening of symptom dyspnea. So we had to give several drugs to avoid those anemia. And yeah, needless to say, all 3 drugs have the fatal defects, so we cannot prescribe these drugs for the pregnant women or the kid-bearing age. Yeah. So other than that, since pulmonary arterial hypertension itself is a fatal disease, so even though there are some adverse effects, we have to use, otherwise this patient would die. Yeah. Thank you.
Okay, thank you. Any other questions? Okay, please. Thank you.
Okay, thank you. Good afternoon, Prof. My name is Muhammad Fadila. I am a master to doctoral student from the Faculty of Medicine, Universitas Indonesia, and I want to ask about the bosentan again. Yeah, as we know that bosentan is very beneficial for the pulmonary arterial hypertension, but nowadays that our patients, especially in the primary clinics, and yeah, they get a lot of drugs like hypertensive drugs or whatever it is. What I want to ask is, how about the drug interaction studies that have been researched so far about the bosentan with other drugs, so we can manage to prevent that? Thank you, Prof.
Okay. Excuse me. There are several drug-drug interaction with bosentan and other drugs are reported. For example, we have many chances to use bosentan and sildenafil both, because both are approved for pulmonary hypertension. And if we use bosentan on top of sildenafil, the effect of sildenafil decrease and the effect of bosentan may increase. But this is the kind of the consequence of the investigation. But actually, in clinical setting, we would not change the amount of the drug. So, and other drugs, as far as I know, or I don't care for drug-drug interaction in clinical setting. Yeah. Thank you.
Any other comment? So Sensei, so it's a interesting journey. You as physicians also and as a scientist. So some of our audience here is really some cardiologist residents who will work at clinical setting. So can you share your experience? How can you do both at excel as a clinician and also a scientist? Even you take long years to found some peptides and until it becomes from the peptide until it become drugs medication, and you did it also in the as a pulmonary hypertension specialist. I think it's very, very rare. Can you share some experience? How can you become a scientist and also a clinician?
Okay, this is very difficult question to answer. But what I can say is that, since I'm not smart enough to do both at the same time, so in some period, I cannot do both. So in some period, in fact, when I was a graduate student, I devote myself into basic research. So I forget about the clinical work. So in these 6 years or so, although I tried to attend the grand round or clinical discussion, but mainly most of my time were used for the basic research. And when I came back from the States and joined the cardiology department, I had to work as a cardiologist. So I cannot use any time for the basic research. So when Dr. Bambam Widiantoro joined us, I seldom appear to the laboratory. So they work by themselves. And so I really missed those research time, but also I have to work as a physician to get knowledge and skill so that would apply for the basic research. So I thought that, okay, this is the time for me to work as a physician. So, and I moved to Kobe Pharmaceutical University because I would like to protect my time for the research. So, honestly speaking, I'm a bit tired of seeing patient every day and teaching resident every day. So I wanted to again devote myself into research. So I moved to Kobe Pharmaceutical University, but in order not to forget the clinical question or clinical updating, once a week or twice a week, I see patients, and I try to keep update my knowledge. So, some are working as a resident, and I think this is the time to work as a resident. But just keep in your mind that if you have some chance to hear some scientific things or read something, then I think those stimuli is very important. And also, another thing that I want to say is that since older people say always same things, that I did not intentionally think those. I just kind of did what I thought it's interesting. So I did not think seriously about the future. So I happen to be this. But since all, so for example, there are some times that you have to choose in your life that this way or that way. Okay, so in this case, I myself always thought that I should choose the difficult way, not the easy way. Yeah. So I chose intentionally difficult way. And so this is all my responsibility. So also I thought that I should not regret my choice. But, if you become my age, and I would not choose difficult way. Okay. So after the age of 60, then you should choose the easy way. Okay. But when you are young, you should choose the difficult way. And maybe in the future, your dream comes true. Yeah. Thank you.
Okay, thank you very much. Just big applause for Professor Noriaki Emoto. So, I think before we end our session, maybe Dr. Bambang want to share some comments, suggestion, or some words. Because Professor Noriaki Emoto always mentioned Dr. Bambang Widyantoro in his slides. Please.
Yeah, thank you, Dr. Hari. Emoto Sensei, again, thank you so much. So, it's so unfortunate that Emoto Sensei, due to the COVID pandemic, we are just be able to invite him again to come to visit our university this year. Actually, after 2019, we have so many plans. We would like to introduce our graduates from University of Indonesia to join his lab to follow my path, Dr. Hari, Dr. Suko, Dr. Sunu. And actually, is there Dr. Adam here? No? One of the graduates from Professor Emoto Laboratory is currently also the residents in internal medicine, Dr. Adam Prabata, the influencer in the Instagram. So I think 3 points that I would like to share based on the lecture from Professor Emoto. So first, you can imagine that in our field, in medical field, in biomedical science field, it is a really long journey since the finding of one small peptide into the clinical application. And a lot of experiments, a lot of studies, clinical trials, fails. I'm the one who still cannot move on why endothelin receptor antagonist is always failed in clinical trials of heart failure. While every single study showing that in every heart failure patient, the plasma level of ET1 was increased so high, it predicts the worst prognosis of the patient with heart failure, but all the trial of endothelin receptor antagonists were failed. Yeah, mainly because another side effect that you also mentioned initially, the fluid retention due to the endothelin receptor antagonists. But currently, I think, Sensei, we have a, a good future since we are now having SGLT2 inhibitors that has a diuretic effect. So I still would like to propose that probably combination of ET receptor antagonist with SGLT2 inhibitor may give beneficial effect for some subpopulation of heart failure, probably not the reduced ejection fraction one, but probably with HFpEF. But we don't know until we conduct some trial. You have any comment for this?
Yes, that trial is undergoing now.
Yes. So yeah, don't give up on something that you believe. That's the first point. And the second one, related to Dr. Hari's issue about being a physician scientist. Of course, it's the hard and difficult pathway. And since we are his student, so we also choose difficult pathway. That's why we become the lecturer in University of Indonesia. Otherwise, we will just choose to do clinical practice in a private hospital. So, Sensei, again, thank you for inspiring us. All the learning point is not just what we have in the end of our study to get the PhD, but Emoto Sensei teach us how to teach systematically, identify the research problem, discuss how is the method to resolve the problem, and he still maintain the time to supervise us. So he worked in the hospital morning to afternoon, and then just came to the lab after 5 p.m. until midnight. So we used to work until midnight. So for you guys, the young generations. So when in your age, so I went to become Emoto Sensei student when I cardiology resident semester 4th. Okay, so I leave residency program and joining the PhD program for 5 years. I forgot all the clinical works, but yeah, I learned a lot. I learned a lot in a good way and in a hard way. So yeah, everything should be with struggle, and yeah, you will find the result years and years later on. So again, Professor Emoto, thank you very much for sharing the experience, and the endothelin is, I think, one of the most successful stories of translational research, how from bench to bedside to be applied to the patient. I think Dr. Sunu, Dr. Suko, Dr. Hari, and me was very lucky to get the opportunity to learn in Professor Emoto laboratory. So thank you again, Sensei. Give applause to Emoto Sensei.
Thank you very much.
So I give back to Dr. Rani to close the session, maybe. Yeah.
Okay, thank you again to Prof. Emoto to sharing the knowledge and also the value of life for our young generation who attend this activity. And then I would like to thank you also our moderator, Dr. Hari Sakti Mulyawan, and also Dr. Bambang Widiantoro to bring Prof. Emoto here and sharing his value and his knowledge. We hope this event will be eye-opening and inspiring to the participants, especially our young generation. Don't forget, choose the hard way when you are young, and then choose the easy way when you are old. Now it's time to choose the hard way. Okay, before close the event, I would like to invite Prof. Emoto to receive placard from Magister Program on Biomedical Sciences FMUI, and then we will take photograph Professor Emoto, Dr. Hari, and Dr. Bambang. And after that, all of us take photograph with Professor Emoto, And then thank you everyone for your participation in this room and then also in YouTube. Please join us for our next guest lecture. And then see you again. Good afternoon. Wassalamualaikum warahmatullahi wabarakatuh.